So let's start the overview. And again, if you have any questions, it would be great to have it in the chat or in the actual chat at the end. So please write it down for you. So a little bit about us. We are a company that started its way in 2017, its commercial way, and we already have many clients around the world. You can see some of the names just here down below. This technology was developed at the Hebrew University. So we are a spin-off from a university development of the product. We, of course, further developed it and made it a commercial product. You can see in our website there are many papers that were already published using our technology. So you're welcome later on to go into the website and have a look at these papers in the reference section. So I'll talk first about the principle, the idea of the system. So the idea is very similar to a smartwatch. We are measuring the dynamic physiological status of the plants, only we don't do it for one plant, we do it for many plants at the same time, and we do it continuously as well as like in a smartwatch. Same idea. We use the parameters that we are measuring, parameters that are correlating directly with yield. So we're measuring traits like transpiration, stomata conductance, stomata activity, biomass gain. So all of these directly correlate with yield. So based on that, we learn very quickly, within days, how the plants cope with different conditions. So we don't have to wait, full season. And also we gather a very large amount of data, precise data. So this helps within days just to get a very good grasp of how the plant cope with different situations. The system is doing three main things. One is control, so the control of soil and water. Each and every plant can get its own personalized treatment. Also at the same time, we measure the plant. We measure the plant and its environment. We measure plant physiology, so different traits that I will share with you in a minute, and also different environmental parameters. All of this being analyzed in a very nice GUI software already with statistics in real time. So you really can see what's happening within the experiment at the time that the plant grows. This is how the system looks, and the basic idea of the system is that we measure the plant traits with the help of scale or sensitive scale. This is the major sensor. So this is here. And we also have atmospheric sensing and additional sensors like soil and plant of different kinds that you can add also. This is our controller. This controller is sitting next to each and every pot, which allows us to provide randomized structure for the experiment and also personalized treatment for each and every plant. Of course, measure all the data, controlling the experiment, collecting the measurements from the different sensors, uploading it to the software. Here we have the exit for different sensors that hook up there, and you can hook up to 4 different digital sensors on top of the least limit or the scale and the atmospheric sensing. So you can see that this is like one unit within a system. So it's an independent unit which is hooked together as one system. And of course, you have all the irrigation components, the electricity, and so forth, the plastics that helps us to regulate the water. You can see here water drainage, so we also manage the drainage. And also we make sure that there is no drop of water that is getting outside of this, or from the bottom or from the top. And the only place for the water to get out is from the leaf. And this is how we measure transpiration, based on the changes in the weight of the pot. During the day, we just let the water to transpire through the plant, and we measure these differences in the scale. Sorry, I will move on. Oops, I see that these drawings continue. Let me just stop sharing and unshare again. And share again, sorry, for this. Okay. Now these are the different traits that we're measuring on the system. You can see in green, it's the plant parameters, and in blue is the environmental parameters. Where we measure main traits are that we measure is transpiration, and this we measure about 500 times a day, so around average of every 3 minutes. Biomass gain once a day, water use efficiency, stomatal conductance, again, ongoing dynamic stomatal conductance, which is usually a trait which is very, very hard to measure. We measure it easily with the system. Root influx, which is root water uptake as well. And also different like atmospheric sensing, soil sensing, and also in the reservoir. And also additional plant sensors like an NDVI and IR can also be added. All of this information goes up to a cloud software, which you can see on the right, some graphs of the software and the nice GUI. You can have already it with statistics, with t-test, ANOVA, box plots, everything already being basically processed for you. The controller, how we control the soil water conditions is with the help of this inside. This is the inside of our controller. You can see two tabs, A and B, and these tabs are managing the water. So it's two different independent lines. You can set drought treatment, salinity, or different water regimes, salinity, nutrients, even flood. We have flood control as well. And you can also set these different, each one of these lines, you can actually set with different controllability. So you can do it either by time, either by target weight, or by a target for the soil sensor, or even by transpiration, because we measure that, and also additional sensors. So you have a very, very nice flexibility on how you want to set your experiments. And you have feedback. It's a feedback system. So when the plants are growing, it's actually taking it into account. This is like a more complex structure, but it's all automatic, so it makes it very easy to do on the system. These are for example, it's an external unit that makes, in this example, 8 different solutions. So we have 2 in the basic setup, and you can have 3, 4, 5, 6, 7, in this case 8 different levels of nutrients or different nutrients, and you can see that you can randomize it on the table. You measure transpiration, you measure with it, so these are just examples of the graphs that were extracted, transpiration of the different line, the different treatment, and also water use efficiency, either agronomic or by the system, using also the biomass gain by the system. So these are different analyses that you can do with what we are measuring. We can provide this for almost, we didn't have any problem to provide it with any plant so far. You can see we have 2 sizes of the system, smaller and larger. Larger for bigger plants like trees and vineyards and others like this, but also a small one that captures like 80% of the different crops being measured on the smaller one. You can see just an example of a system, how it looks. This is the Tvacheningen, and this is for, it's used for, in this case, for quinoa study, quinoa breeding, but that was done in salinity and drought conditions. But you can have, and this is being used for many different type of experiments which are being run in parallel on the system. And this is in the NAPPN facility, new facility for phenotyping. This is another example, again, just so you can envision it in a real environment. This is UC Davis, managed by Mars Wrigley, for the cocoa breeding program. And here you can see a video about, I hope it will work. Yeah, how it's actually measuring while the plants are growing. You can see the graphs, and the plants are growing within 1 month of a trial, drought, recovery, everything is being captured and being analyzed. This is how the analysis is being done. This is just one treatment, in this case it's genetic line, in drought and recovery. And you can see that we can control the bottom line, so the soil water content is being controlled, so the drought is actually very nicely going together. And on the same time, we measure the different lines and their response, either in drought or in the recovery of the different genetic lines. And this is all done by the system. Here you can see within a day view, so you can see from 6 a.m. to 6 p.m. and how the plants are responding to the environmental conditions. The same genetic, in this case, tomato genetic lines. And you can see very nicely how the GS, the stomatal conductance, is responding very nicely to the pattern of the light, and transpiration is actually following the VPD conditions. And you can see it even by the eye. But you can see here the difference between the lines within a day. So you can really capture a momentarily reaction. For example, if you look at this graph, you can see the blue line very nicely. The blue line has a very nice lead on a certain time of the day. We call it the golden hour. And this is the time where the plant is the most productive. The system can detect that very easily. So what applications does this apply for? So abiotic stress, screening of different kinds, biotic stress as well for early detection, because the physiological traits are very fast to react. So usually it's around 5 minutes reaction for the plant to change its, to apply its reaction. And we measure every 3 minutes. So you can really capture every change in the plant reaction. So what we're also, this is applicable for, of course, physiological studies, but also nutrients and bioag screening, seed treatments, and optimization of growth. How are we different from other solutions? So, of course, for manual processes, we're very different because we are automatic. This solution is automatic, very precise, very fast, not being done plant by plant, but all the plants feel the same environment, the same conditions all together, and they're measured at the same time. All the plants, it could be 500 plants, and they are at the same time being measured. Depends on the size of the array. It's not measuring on a leaf level, it's measuring on the whole plant level. It's not destructive. It's of course comparable because we measure it at the same time for all the plants. And you have also this automated, very flexible automated irrigation control. If you look at digital devices that you hold by hand, for example, for physiological measurements, still you have a lot of these hurdles of the slowness and the heavy workload, the plant by plant on a leaf level, which is not comparable then at the same time. And without any irrigation control, this still sticks out, so it's still the same. So with our system, we actually overcome all of this altogether. Also, if we compare our system to imaging, it's very different. The imaging is looking at the structure, the color. It takes time for the plant to change structure or color, where the physiology is immediate response. It's the function of the plant more than the structure of the plant. It's continuous measurement and not just one or two times a day. And you can capture immediate responses by the plant. And also we have our flexible control abilities that I demonstrated before. So again, coming back to the same idea of the smartwatch, or similar idea. That's it. I will stop sharing here, and I will be happy to have discussion and any questions that you might have.